Welcome to Revolution Against Evolution. I'm your host, Doug Sharp.
Your co-host Rich Gear here as well, and well, Doug, we talked archaeology last week. We're going to talk some enzymes and proteins, and that's your bailiwick.
Well,
Really
yeah, that's something that I really enjoy talking about because it's something that got my motor running when I was just in college and high school, actually. I conducted some experiments trying to create life in the test tube.
The famous sticky goo experiment. I love that.
And so now that I know a little bit better, because of what I learned in chemistry and in biochemistry and biophysics and all the courses that I took when I was in college, I'm going to explain a little bit of this stuff to you. We're going to talk about proteins and enzymes explained.
And I like that because I've heard it explained, but I'm a guy who needs about 3 times before it finally sinks in to how it actually works. I still
Well, I've explained this to you several times,
and I still haven't got it. See what I mean? Some things just don't get in there. You know, you know, Doug, I don't understand. Like, even some subjects that I really like I love geography and I love astronomy, but nothing sticks. Now dates, boy, they stick in my head. I don't know why dates stick in, but this stuff is another one of those geography, astronomy situations. I'm interested and it fascinates me, but I still need to get explained, especially if I haven't heard anything about it in 6 months or 8 months. So,
well, we're going to get a little chemistry lesson here.
Good.
And it's something that has been my passion ever since I was given a chemistry set as a young boy, and I was about 12 years old when I got my chemistry set, and I wasn't the same afterwards.
maybe that would have helped me. I don't know. I never got one of those.
But they don't have those anymore, unfortunately.
No, because kids might eat it or something. Or
Yeah, it's some of the chemicals in that chemistry set was actually fairly dangerous, you know. In fact, I ingested some by accident. I found out.
Is that what happened? No, I mean,
But I remember that I was playing about with some of the chemicals in it, and there was one calcium hypochlorite, and so I mixed that with an acid. And I just noticed a strange smell, and I said, "Ooh, I don't know what this is," and so I captured some of it in a container. And I took it to my chemistry teacher in high school, and he said, "Oh, it's chlorine."
oh yeah,
And
that'll
so-
kill you. Yeah.
And so it was a little bit of a lesson. That's why you don't mix bleach and vinegar or ammonia in your washing machine, because
Yeah.
that's what will be produced. Well, you don't want ammonia in it anyway, but
I was just saying, well, who would put ammonia in your laundry? I don't know. I don't know. You never know what works, but okay. So then,
anyway, I learned a lot of the chemistry, and one of the things that really fascinated me, there was a book by Linus Pauling, and it was called The Nature of the Chemical Bond. And and he had a way of explaining this stuff that was really very simple to understand. And this was a fairly complicated concept to convey. He was able to show how the different atoms have these, especially carbon. Carbon has these 4 different bonding mechanisms that you can have a single bond, or it's a covalent bond. They share electrons. They pass them back and forth.
that's what covalent means. Sharing.
Yeah, you're sharing the electrons. They're sharing an orbit, and it's orbiting around the 2 atoms.
Okay.
Now it can have a double bond, and then you have another orbital that goes kind of like this on both of them, but those can be shared, and so that doubles the bond, and you can actually have a triple bond. And if you have a triple bond, you have some things like acetylene, which is a pretty dangerous chemical. Yeah, I use
Yeah.
a acetylene torch, you know.
Acetylene torch, I remember using those, yeah.
And cyanide is a triple bond between a nitrogen atom and a carbon atom. And so those, you would think are fairly strong bonds, but yet because those things are fairly flammable, those secondary bonds that break down a little bit.
Are they what you would call a weak bond or something, or are there things
Well,
called strong bonds or weak bonds?
This is a strong bond, and if you have a triple bond, it's a strong bond altogether, but the parts that are extra are the ones that break down fast. So,
Okay.
So it's a really, you have strong bonds and weak bonds. There's another kind of bond which is like between a metal and a halogen, which is like chlorine. So you have sodium, which is a metal, chlorine, which is a halogen, and you have a positive charge on the sodium and a negative charge on the chlorine.
Okay, you said, wait, the chlorine steals it from the sodium, is that what happens?
That's right, yeah.
My
So
chemistry
then,
major, you know.
then what happens is that you get a nice strong bond, which is the ionic bond, and it attracts each other, like electrostatic type of
Okay,
thing.
okay, like a magnet or something.
Yeah. And then you have, and then there's things that, and these take place within proteins and in other molecules, but proteins especially, these are very important in understanding how they operate.
Yes, because we've got, you said proteins and enzymes explained. Now, enzymes, I think they're kind of the building blocks for proteins.
Enzyme is
Or
a
not.
special kind of protein.
Oh, enzyme is a protein.
Yes, enzyme
Ah, I
is a
never,
protein.
okay,
And, but not all proteins are enzymes, okay?
okay, but all enzymes are proteins.
That's correct,
Okay,
yes.
all right,
And the enzyme is a special purpose protein for the purpose of like breaking down food or catalyzing reactions in the body.
didn't know that.
It's sort of like, for example, if you eat your dinner, you're going to have enzymes in your stomach and elsewhere in your body that will take the particles of food, and there will be like a little a key and lock mechanism that gets manufactured in the body
That's fascinating,
that grabs onto this particle and bends it right into the correct shape so that it will break down.
It bends the food or?
It bends the food.
Okay, wow, okay.
And it actually conforms into the shape of the food, and it actually has some intelligence behind it, so it does it on the fly. And this is actually something that happens with antibodies. Antibodies does the same thing, where it has this lock and key mechanism, where it recognizes what's out there, the bad germ or whatever is out there,
Right.
and it manufactures this little section of it that matches it, and then it'll attract it and spirit it out of the body.
Yeah, okay, I've
And
seen
so-
diagrams of that, yeah.
So that antibodies and all these different things are really special designed things by God that He put in there, and it actually operates in an intelligent manner.
So let me get just, so enzymes are proteins, but like let's say you're eating meat and that's a protein already.
Right.
This protein does something to the protein you're eating and shapes it so that your body can make more proteins or more what?
Yeah, it'll break it down into its components, and then it'll reuse it elsewhere in the body to put back together.
The ultimate recycling mechanism. Huh?
That's exactly what's happening.
Okay,
And
all right.
there's another kind of bond which is a little bit different. It's called a hydrophobic and hydrophilic bond. And what it is that there's parts of the proteins that are like oil, and it's usually a oily type of side chain on the amino acid, and it doesn't like water, And so if you find a bunch of those next to each other, they're going to sort of bend it so that they're next to each other, so that they're away from the water.
You know, okay.
But there's the hydrophilic ones, the ones that like water. It actually has an OH on it, which is a part of the water.
Is that, yeah, oxygen, hydrogen?
Oxygen and hydrogen.
Okay.
And so because it has the oxygen sitting there, it will attract water, and it sort of likes to waft out in the water and like it. And so what that will do is help bend the molecule into this shape. There's another kind of bond called the van der Waals bond, which I don't think a lot of people understand. It's very weak, but it's enough to actually help shape things.
So all these are used to shape proteins, or
Right.
to shape whatever it is that they're working on,
Right.
to make proteins, or to feed proteins.
This is all part of that lock and key mechanism,
Okay.
where it'll match the opposite of what it is on the food, so it will bend it into the proper shape to be broken down. And the fact that it is weak bonds helps to break it down. And so I find that really fascinating, how this all works together. I saw that there's something we call a substrate. A substrate is what the proteins are working on. They sort of get in this lock and key mechanism, and what will happen is that once there's enough of this new substance being made by the enzymes,
Okay.
it will actually bind onto the enzyme itself and
Okay.
force it into a different shape, so it shuts down the process.
Okay, so it only goes so far.
Yeah, it's like a servo mechanism where you have this whole product of this mechanism shutting off the whole.
And when you get the thing into the shape or the protein you want, does this create energy? Does it help DNA
Yeah,
process?
that's all-
I'm
all
just
part of
curious
it.
what okay.
It's all food breaking down and making it possible to manufacturing new stuff in your body.
Okay.
And so what you ingest into your body, and that's why it's really interesting that we have all these different kinds of foods that you eat,
Yeah.
and your body will adjust and adapt and recognize what those foods are, and then break them down.
Now let me ask you this question, Doug. The enzyme works on the food and bends it into shapes so your body can use it for different processes. How does it know what different process that it goes to? Is there something? Is that part of the mechanism too, or I don't want to get too far afield, but
I'm saying there's some sort of vitalism or intelligence behind all of this.
yeah.
Because it's really part of the whole mechanism of the body, and even the lowliness gnat, or can you imagine? You're an engineer, and you are assigned the task of creating a flying machine the size of a gnat.
No.
Okay.
That's a nasty problem.
Yeah, but
Sorry.
that is really what is part of all of life, even from the lowliest bacteria on up to humans, through the zoo, like
Yeah.
they are all intelligent.
They're good to you via the zoo, isn't that the glib saying?
And the thing that it is that if you understand that all of life has this background mechanism built into it, and it's all complicated, it's all beautiful, and like I thought I'd give you a little idea of the 20 amino acids that are.
Yeah, I was reading that the other day, and I went, holy man, I didn't know there was this thing.
There's 20 amino acids, and they're made up of a structure where there's a lattice work that's all the same for all of them. but there's a side chain that makes it unique, and then it has a acid part, which is a carbon, oxygen, and hydroxide part. And those parts are on opposite ends of the amino acid.
Is that always the way it is?
Yeah, this,
Okay.
well, there's an exception, I'll tell you.
Okay,
But
yeah,
the alanine is one of the simplest ones, and it's just got the amine part and the acid part, and it's connected with together with a side chain. The glycine is the simplest one. It is just one carbon and the amine and the acid part.
wait, I thought you said that, wait, so alanine is not the simple,
The alanine is almost the simplest one. The glycine is the simplest.
okay,
Arginine has a long chain. That would have been one of the ones that it's actually, that would have been more of the hydrophobic one. Cysteine is naturally occurring, it's got sulfur in it. Now the sulfurs are special because the sulfurs bond together and they'll connect chains together, and they'll have, and that's a strong bond, is the sulfur bond.
okay,
Tryptophan is the one I studied in high school and in college, and that was one of the more complicated ones. It's got a double ring in it, and it's really, I found that if you do to it what I did, was to take a electric arc
yeah,
and try to zonk it and see what would happen, and it basically turns brown into some gunk that
tar, goo.
well, it turned the solution brown. Now, I made the mistake of doing it in water, okay? I found out that if I'm trying to combine amino acids, water is an enemy to that reaction.
Oh boy,
So
the
I have
very
to-
thing they're looking for life on another planet, water,
Yeah.
does not makes amino acids not work well. But I think you said there's some that like water, some amino acids that like water. This is one that doesn't like water?
Right.
Okay.
And the typical thing about tryptophan and tyrosine, which is the other one I worked on, they have these big aromatic rings, and they just take up space. They sort of hang out there, and they, because of they're big, they occupy that space. Now, although because they're big, they're very difficult to actually create. The origin of life experiments, they don't find either of these as outputs to them. Proline is one of the exceptions I told you about, is that the mean part is part of a ring. And again, that has its own little personality of its own. Glutamine is a amino acid that's one of the hydrophobic ones. Histidine is one of the exceptions that has the amine part as a ring.
When you say rings, you talk about these things that connect like in a circle?
Yeah, in that case, there's a pentagon. The more common one is where you have a hexagon.
Okay.
And in these cases, these also have, what do they call it, a aromatic characteristic, where it's actually a double bond that sort of goes back and forth. It's the whole, because it's a ring, it just goes round and round, and shares the electrons around the ring.
Okay, all right. Yeah, I just always curious, because they're always drawn kind of like with angles. I always think of rings as circles, so.
Yeah, right.
Okay, so that clarifies that for me, okay. So then you got like these ones, they're not, this next one.
Yeah, isoleucine and leucine those are hydrophobic. Lysine is also hydrophobic. Methionine has a sulfur in it. And if you ever burn some of these amino acids, especially methionine, it'll release the sulfur, and sulfur stinks.
Nah, sulfur does not smell good. Yeah.
Phenylalanine has a ring on it. The threonine is a hydrophilic guy.
I was wondering when you needed one of those, I like water.
And valine is a little bit of a hydrophobic as well. So it's mostly the hydrophobic ones that do the shape, because they like to congregate together and they'll bend the.
Okay.
Bend
Are these
amino
all the amino acids? Or
Those are all the 20 amino acids.
okay.
And so
They've only
then,
found 20. So they've only found 20 of them. Is what's happened? Is that what it is, or how does that?
there
work?
are more amino acids than just the 20, but these are the ones that are used commonly in life.
In life. That's what I wanted to clarify. Okay. Okay. Good.
And I've talked about this before, but when you create these in the test tube, you get equal mixtures of a right-handed and a left-handed variety. But life only uses the left-handed variety.
Right. We don't.
And so I've gone through that before, where the odds of that happening are just not believable.
Yeah, they're astronomically low.
Yeah, I talk about
The snail.
the tail of a snail,
Snail.
whose job it is to move the entire Earth one molecule at a time over to the other side of the universe and back.
Yeah.
And
Go ahead and tell the story. It's not. It's worth. We got time.
the snail goes at a rate of 1 millimeter in a million years.
Oh my God.
And so you have a protein factory whose job it is to produce a usable protein and you produce new proteins at the rate of 400 tons a second. And by the time you fill the universe up with these proteins, the snail will have done its job several times, and obviously, then if you get one protein that works, well, you've got to go get a whole bunch more in order to get just one viable cell.
Yeah. Or yeah. Well, and even the organization of a cell. See that's the thing. Back when Darwin proposed his theory, the cell was. It basically looked like a blob of jelly, or it was just kind of an amorphic kind of a. They didn't realize. They had no idea what. We've taken the cell apart before. But even in biology class, you look at it, and you go, cytosomes, cell walls, and mitochondria, and all this stuff that's inside there, and you go, wow! And they're all machines.
Mm-hmm.
They're all incredibly complicated machines
Right.
that do things. It's just really, really a cool thing. I like some of these processes you put in here, Doug, and I remember doing some of these illustrations, and I went, wow, because I never really understood. You got transfer RNA, messenger RNA, and you got all these different kinds of RNAs, and I never quite figured out what the RNA, what that was, that did something to create the DNA, but it's proteins. It's all these things that are all interconnected to make the life work.
Well, let's talk about the DNA first.
Okay, let's do that.
We'll just start with that because, of course, there's going to have to be a process that creates it. And a lot of the things that happen when you're creating the DNA, well, it's being replicated from something else. It's being replicated from another pattern. But there's a protein called the DNA polymerase, and its job is to encircle the DNA to make sure it doesn't twist up or that it's being folded in the proper direction so that it doesn't fall apart, or doesn't kink up while you're making it.
Okay.
And so what happens when you, but there's the DNA codes for RNA. There's 3 different kinds. There's a messenger RNA, which is what transfers a message to a ribosome. There's the transfer RNA and the ribosomal RNA. And the ribosome is a big blob where all the magic stuff happens.
I know.
And there's like 3 of these nucleotides in the sequence, that code for a protein. And there's on the transfer RNA, it reads this, and it connects to the messenger RNA here. It goes along it just like this, and it reads it, and the transfer RNA picks up an amino acid and adds it to a growing polypeptide, which ends up being the protein. And the rRNA, the ribosome RNA, is just part of the ribosome. That sort of keeps it into shape.
Okay.
And so then you have that growing polypeptide, and you have at the end a completed enzyme, and it folds it up in the proper shape, just like it's supposed to do, and then it's active. And like I said, if you bend it out of shape, it doesn't work.
It won't work, okay, yeah.
If you try to create this, just artificially, it doesn't work.
Okay.
And so then, here's the chicken and the egg problem, is that this completed enzyme will come over, and one of those would have been the DNA polymerase.
Okay.
Well, which came first, the DNA polymerase or the DNA?
Yeah. That's it's one of those things where everything has to work or nothing works, correct?
Exactly, yes. And so that is really the question, is you have to then have an energy producing mechanism that helps regulate this. You have all these other
But then,
things.
this is, how do evolutionary people, biologists, who know what you're telling me,
Right.
how do they, what ad hoc explanation do they even come up with to explain how it could work?
Well, they assume there's no God, and so they have to figure out a way to get around that.
But have they done that?
That's the real trouble, is that they really haven't done that. They don't know how to do it, and so the question is, a reason, and so they speculate, well, we had life imported from outer space, or there's...
panspermia, which basically begs the question, because they have the same, ultimately you have to get to the uncaused first cause, which is no old theological argument, but it's still valid, because I'm sitting there going, do they even have an ad hoc explanation when you look at the complexity of the machinery at the microscopic level? And if one part doesn't, one part needs the other part needs the other part. They won't. How can and I can't even imagine even a naturalistic mechanism that could have created these things at the same time without an intelligence behind it, Doug.
No, believe in God. That's the easiest way.
Amen.
We'll see you next
Amen.
time on Revolution Against Evolution.